Hi, my name is Christina Ferroni, and I'm a PhD candidate at Queen's University. And today I'll be discussing the appraisal of a vendor-supplied NGS filter. I have no conflicts of interest to disclose. So first of all, NGS is being used clinically more and more in MDS, but there aren't currently standardized methods with a variety of different platforms, targets, and variant calling pipelines being used at different sites. We previously validated the Oncomine myeloid assay, choosing to apply the vendor-supplied filter and pipeline clinically. However, we noticed during the validation phase that not all relevant variants were captured by the pipeline. We then decided to look outside of the vendor-supplied Oncomine filter to explore what variants with potential clinical relevance are not being captured by this filter in a cohort of 74 patients with MDS or MDS/MPN. So what we found, as seen on the left, is that 169 variants were detected in total, 63 of which were missed by the Oncomine filter. 42 of these had at least one hit in Cosmic, with 8 of those being found in Cosmic hotspots, all with VAFs below 5%. On the right, we can see the number of detected versus missed variants based on affected gene. Interestingly, the genes on the left with the greater percent of variants missed tended to be in genes that are less commonly mutated or that are more poorly annotated in ION reporter, despite variants in these genes still being clinically important. Serial sequencing further highlighted some challenges of the Oncomine filter, as illustrated by the case of PMDX50, a 74-year-old male with MDS and 4 serial NGS results as shown. Outside of the Oncomine filter, we found the following variants, but when the filter was applied, the highlighted variants were missed. This is especially important because the clonal picture from May 2019 would not have suggested the emergence of a new subclone with proliferative mutations, whose detection may have impacted care earlier on. In this case, potentially starting azacitidine treatment months sooner. This case example also highlights the value of serial sampling in more inaccurate variant calling. Moving forward, we still need to assess the more recently released Oncomine extended filter that appears to capture a greater proportion of variants. It would also be interesting to see whether similar patterns emerge with vendor pipelines for other platforms, which we have yet to explore. I'd like to acknowledge and thank our funding agencies and collaborators in the molecular lab at KHSC that contributed to this work. Thank you.